All right, well, let's get started. We'll start off with some introductions. What I'll do is I'll just to make this manageable, and it's a pretty manageable group, so it shouldn't be too much of an issue. But I'll introduce myself, and then I'll just call out each of your names. I'm just going to go down the list as they're ordered here in Zoom, and just take a half minute or so to introduce yourself. So my name is Jeff Spraggins. I'm the moderator for this breakout session. And I am in the Department of Biochemistry at Vanderbilt University. For the past 10 years or so, I've been working on developing technologies for imaging mass spectrometry and spatially targeted proteomics. And I'm currently one of the PIs on our tissue mapping center that's part of HubMap. Raf, you're just next in line. Why don't you go next?
Okay, my name is Raf van der Plas. I'm a Belgian who is actually affiliated with two places in the world. One is in the Netherlands, in the Delft University of Technology. They're primarily computational analysis, mathematical engineering. And I have a second affiliation at the Vanderbilt University in Nashville. So I have an adjunct position in Nashville. So that's where most of the more wet lab-y instrumental work happens, and most of the computational work in our little chunk of HubMap, together with Jeff, happens in the Netherlands.
Next up is Elizabeth.
Hi, I'm Elizabeth Neumann. I'm a postdoc at Vanderbilt. I primarily do imaging mass spectrometry, but I have a foray with antibody-based imaging approaches.
Mike, you're next on the list.
Hi, I'm Mike Lucero. I'm part of 10X Genomics. I'm an industry veteran. And what you see in the background is my backyard in California.
Nice. Rita?
Hi, everyone. I'm Rita Strack. I'm actually senior editor at Nature Methods. So I have been jumping around a little bit between sessions. I handle mass spec imaging, so I just thought I would see what you guys are talking about today.
Fantastic. Stephanie?
Hi, Stephanie Morris here. I'm a program director at the National Human Genome Research Institute, and I'm involved in a couple of programs. One is technology development, and the other is ENCODE. And I'm also sort of jumping around to see what people are talking about. So thanks for having me.
Great. And then lastly, Kavya.
Hi everyone, Kavya Sharman here. I'm also at Vanderbilt. I'm a second-year grad student. I'm doing a lot of work on the computational sides. I'm working with imaging mass spectrometry and different immunofluorescence data sets. Some of my work is on segmentation as well as some proteomics work.
We actually had two people just join, so I'm going to let them connect their audio. For those of you that just joined, we are just doing some introductions right now. So Cody, are you connected? Are you able to introduce yourself?
Yeah, can you hear me?
Yeah.
Yeah, I'm a graduate student in Ken Lau's lab at Vanderbilt, and just trying to listen in for a little bit.
Great.
You guys are up to.
Perfect. And Stephanie, do you want to introduce yourself? Or are you just bopping around checking out how things are going?
I think I've already introduced myself. Stephanie Morris?
Oh, I'm sorry. No.
It's okay.
Who just jumped on?
Maggie?
Yeah. Sorry. Maggie. Sorry.
Yeah, I just came in on the facilitator. I'm just checking to make sure you're all connected and you're underway, and I'll leave you alone otherwise. And you know where to find me if you need me.
Perfect. Sorry, the names are jumping around in Zoom, so I got out of order there. Okay, so in terms of setting up this breakout session, we are kind of Vanderbilt-centric in this group. So Mike will probably be pinging you and some of the others that have joined to kind of see what we're up to see what ideas you might have and how we can do things better. But in terms of the breakout group kind of mechanics, if you're going to be a passive observer, although it's a pretty small group, so I might just call on you, you're supposed to turn your camera off. The moderators, I'm going to do my best to just kind of facilitate the conversation. One of the things that I do need help with are identifying the scribes of the group. And they've asked us to do 2, which that's a pretty high percentage of the folks that are in the room right now. Would anybody like to volunteer for that activity?
I can scribe.
Thank you, Elizabeth. Anyone else for the second scribe? I can take notes as we go along too, so maybe I'll act as the second scribe since we have kind of a small group here. Okay, now with that said, does everybody have access to the Google Doc? Let me make a copy of that and I'll put that in our chat box right now, just so everyone does have access to that. If Elizabeth's going to be the primary scribe, and I'll take notes as we go along as well, but all of us can edit this document and add anything to it. So if we remember from the last time around, basically we're going to spend this first 45 minutes kind of describing some problems and some things that we hope to be able to collaborate with, although a lot of us all already work together, so already kind of collaborating. But at least we can identify some challenges that we can seek some outside help on. I guess one of the things that might be nice, Mike and Rita and Stephanie, you know, how much experience do you have with spatially targeted mass spectrometry? Basically, is it valuable or interesting to you all for us to kind of do a brief technology review of what is available?
I have none, and I was really meant to be just an observer.
Okay.
Yeah.
And the same with me. I have none at all.
Okay.
This is Stephanie.
Great. So the way, I'm just going to give a brief overview of where the technology is, just so we're all kind of on the same page. So we tend to think about the spatially targeted mass spectrometry technologies, and they kind of fall into two categories. The first of which is imaging mass spectrometry, or mass spectrometry imaging, depending on where you are in the world. And that is exactly what it sounds like. It's actually generating images using a mass spectrometer. Now, this isn't the antibody-based systems, whether it's MIBI or CyTOF imaging or those technologies where you're applying metal tagged antibodies and using a mass spectrometer to detect those metal tags. This is using a mass spectrometer to detect the endogenous molecules, so metabolites, lipids, peptides, proteins within the tissue. So you're generating full mass spectra at every pixel location. So we're generating pictures in that approach, in the imaging mass spectrometry based approach. And then secondarily, you can think of the other way to do a spatially targeted mass spectrometry experiment is in what we call profiling mode. So in that case, you're not necessarily generating a picture, but you're sampling in a way that's spatially targeted. So a lot of these systems, those kind of fall into two primary categories. The first is like a laser capture technology, or sometimes physical capture. So in those cases, you're kind of cutting out a small region of tissue and then processing it for proteomics or metabolomics or lipidomics of some kind. Secondarily, you can use liquid junction. So you can use small droplets or small devices that allow you to flow liquids over a surface so you can extract the endogenous molecules. Now, so that's a very high level overview. In terms of the imaging, there's all sort of sources that you can use to generate data and images. We're going to focus primarily on the laser-based approaches because that's what the majority of the field does, and for all the people in this room that are doing imaging, that's what we all do. So we'll probably focus mostly on matrix-assisted laser desorption ionization. It's a laser-based approach. And like I said, it's probably the most widespread of the imaging based technologies. Are there any questions about that as we kind of move away from the lecture portion of this meeting and more into an open discussion? No.
No.
Great, we're all good. So, so this is where I think it'd be good for us to kind of open things up a little bit. I've set up, if there aren't specific questions, which there probably doesn't seem that there likely will be, I've set up a few guiding questions and topics for us to talk about. So these include things like sensitivity, spatial fidelity, how we're aligning our data with specific cell types, quality control, and then how we deal with non-biological variants in our experiments. So does anybody have a preference on what we start with? I know this is mostly our group, and Cody, please jump in if you've got specific things that you're interested in. I know you're really interested in learning about these technologies specifically.
Yeah, sure.
Any takers on where we're going to get started? Well, I think in terms of breaking down, the most interesting thing, and probably one of the most pressing things we see from HubMap side of things, is how do we align our data with specific cell types, and how we're addressing that. I know, Cody, that's something of interest for you all. It's one of the things that we see as one of the biggest hurdles in HubMap in terms of integrating data with some of the other technologies. And so I think maybe let's start there. So for anybody who's an active participant, do you want to help identify some of the challenges associated with linking our imaging mass spec data to specific cell types?
Well, Jeff, I can chime in for a second. So in many cases, I mean, it's a matter with all these multiple modalities of many varieties and many omics to, at some point you want to connect the dots, and the granularity with which you can do that is going to determine how fine-grained your analysis can be. So the finer is the better. Now, until we were able to do some of these things in a spatial domain, it was just an organ or some chunk of tissue, and then it got homogenized, and then yeah, you lose basically any location. You can just say, oh, okay, it's some sample from this or that, and that's our link to link things together. But because the signal to noise ratio is such that the finer you can make that little volume, the more precise your analysis can be. Our continual striving is to make either pixels smaller, to make acquisition points smaller. But of course, none of these things matter if you then can't make that connection. So I think how we do the GPS of tissue is going to be very important, because you need a coordinate system to tie things together. And that doesn't necessarily need to be some sort of Cartesian coordinate system of 5 steps up and 3 steps right. It can be something more rough, but just a little bit more specific than just full-blown homogenization. And I'm sort of interested to see how they are tackling this in the spatial varieties of transcriptomics, because we are currently tying lipidomics, proteomics, peptidomics, anything that wants to fly in a mass spectrometer. We are tying that to either an organ, a subpart of an organ, or even more specific, a cell type, or even more specific, a specific cell type, or a particular structure. But how is this problem being approached by the spatial varieties of transcriptomics? Because on the proteomic side, we have it sort of, we are trying to get as much meta information labeled down there, but I have no idea what is going on in transcriptomics world.
I think.
Oh.
Sorry, go ahead.
I can speak to that a little bit.
Yeah, go for it.
So like if you look at the beginning of like single cell transcriptomics, they did most of their linking with immunocytochemistry. And so they would stain a cell, know what the cell was, and then extract the RNA for transcriptomics analysis. And so they would rely on that as like the gold standard of cell typing, and then go from there and then develop their own transcriptomics like classification scheme.
Yeah, I can second that. So, 10x Genomics, we sell the spatial transcriptomics product. We've built a set of fiducials around the capture area. And what we are moving towards is allowing people to do immunofluorescence measurements, and then do the spatial transcriptomics analysis second, and then being able to align the immunofluorescent results to the transcriptomic information.
And Mike, that's happening in the same sample?
Yeah, that's happening on the very same, exact same tissue slice.
So you don't really have registration problems in that case, since it's all happening in one slice.
No, we don't, because of the fiducials that are built around into the product.
How many fiducials do you use, and is that something that? So, and the reason why I ask is like this is something that we do quite often. Is like, we tend to work with serial sections, so it's a even more challenging problem. But it is a kind of question in the field for the experimentalists. Is, how many fiducials you use, how accurate, how precise is your registration? with a set number of fiducials?
I don't know the exact number of fiducials. It's actually a whole frame. There are hundreds.
Yeah, many though.
Yeah.
Okay. Yeah, so on the imaging mass spec side, one of the things that we often do is that we're typically working with serial sections. And so it's a little bit different than your case, Mike. And the reason why we do that is just because usually we're trying to optimize the sample preparation and sensitivity for each of the platforms independently. And in many cases, that means the sample preparation is totally different. In our lab, we've developed some strategies that, and one, it's kind of similar that you end up with many fiducials, effectively many fiducials, to align one data set to another, but effectively we're using things like autofluorescence microscopy as a way to drive our registration of the two serial sections so that we can connect one data set to another. And what I mean by that is we'll collect an autofluorescence scan before we do our imaging mass spectrometry analysis on the tissue section. And then following the imaging mass spec analysis, the tissue is still present. We can go back and collect a secondary autofluorescence scan before we wash off the matrix. And for everybody who isn't a multi-expert, we apply kind of a small molecule to the surface to do the analysis. So we can see where every single laser spot, fired on the surface. And then we can map all of our pixel locations where the laser actually interacted with the surface to a theoretical pixel map, and then relate that to the microscopy directly. So what we end up with is effectively hundreds of thousands of fiducials because we know where every laser position was fired onto the surface. And we use that to kind of drive our registration between the imaging mass spectrometry and things like microscopy that's collected on the same sample, and then we use microscopy to microscopy registration to go from one tissue section to another. So we definitely take an approach that is kind of a combination of experimental considerations and technologies, and then also some computational tools to drive that registration automatically. Now, one of the things that the reason why I think, linking imaging mass spec to cell types is so important, and this is kind of new for our field, but it seems to be the primary language. We can only speak from our experience within HubMap, but cell types seem to be the central language that will connect all of the technologies together. Because definitely on the transcriptomics sequencing side of things, everybody is always concerned about cell type, cell type, cell type. And then on the immunofluorescence side of things as well, it's all about what cell types are you labeling. And so it really seems to be the main glue that's going to connect all of these data sets together. Now, we're a little bit unique because we are not targeted at all. So we basically, for instance, in a lipid imaging experiment, we're going to detect hundreds of lipids from a given pixel location, but you don't necessarily know what those molecular signatures mean in terms of what cell type they're associated with. So we have to, through analysis and data integration, determine how those signals are associated with different cell types. Raf or Kavya, do you want to speak a little bit to how we're approaching that process?
Yeah, absolutely. So I think on the kind of the topic that you were talking about, Jeff, the cell type differentiation, we're currently using multiplex immunofluorescence for that, that we are switching over to Codex. So a lot of that is antibody-based cell types. So the thing that I think would be interesting to talk about is the way the transcriptomics field versus the more of the antibody field, how they classify cell types, because I think making that bridge is going to be one of the key things in linking these two data sets.
Yeah, absolutely. And one of the underlying technologies for, not only, of calling a cell a certain cell type is the segmentation pipelines, which I know you're working a lot on right now, Kavya. But those technologies and getting a handle on that, the segmentation side of things, so that we know which pixels are associated with cell, is an extremely important factor in us being able to make these connections.
So if I may just make have a question on that. So for the transcriptomics side of the world, sort of, what is the level of granularity that they're striving for? Is it because you mentioned, Mike, that it was down to cell types? So not down to individual cells, or is there a movement towards this, or what's going on there?
There is a movement towards single cell with spatial transcriptomics, but it's mostly through means like merfish and seqfish.
And is this like, if these instruments from 10X, for example, what do they support? Is that something pie in the sky, or is that something that's quite close to what the industry is making?
We're near single cell. You know, anywhere from, it depends on the cell type, 5 to 20 cells per spot, I guess, get analyzed, and we have 5,000 spots per tissue slice. We've said publicly that we're moving towards finer resolution, but we haven't said exactly when. So it's just something we're working on. I think, but I mean, just as a, and so now I'm not talking for 10X, but I think the merfish and seqfish, those methods are actually going to get to single cell first, and they'll end up using the transcriptomic information from single cell, dissociated single cell, to find the marker genes to do their analysis and identify cell types at the single cell level. One of the advantages of those merfish and so on is that you can you don't destroy the tissue when you use those. You can then maybe you could put them into your Malditovs.
Yeah. For us, one of the things that it can go both ways. Elizabeth, you have some experience doing transcriptomics and imaging mass spec on the same samples. I forget which one is better to go first.
So it actually, so it depends. We've gotten it to work most consistently doing IMS first, but it's because the MALDI matrix actually serves as a preservative for the transcript, or the transcript, and MALDI isn't actually that destructive. And so if you do transcriptomics first, you basically trashed a lot of your metabolite information and can't get, I was trying to do small metabolite imaging, and so you actually couldn't get any of that information. You could get lipids all right. But if you do IMS first and then ship it off, you actually preserve the transcripts with the MALDI matrix.
Yeah, and I guess as long as your laser energy isn't too high, then the tissue's intact.
Yeah, and we were doing it on single cells too, so the tissue's more robust than a single cell is as far as damage is concerned. Although we've analyzed broken cells with transcriptomics, as long as the transcript's okay, the cell can still be damaged and it's fine. We usually only get the surface anyway, or we only damage the surface, which isn't where most of the RNAs are at.
Interesting. Yeah, so I guess one thing that would be nice to come out of this, so one of the things that we're kind of tasked with are coming up with these statements, how might we act? And really all of this is about establishing collaborations. Now I know a lot of the people aren't necessarily in the room right now, but I think what would be good for us to try to come up with here are a couple of statements like this that we can then reach out to either people in HubMap or people in other consortia to try to identify points of collaboration. So in terms of integrating transcriptomics, and Cody, I know you're really interested in this, in integrating transcriptomics with imaging mass spectrometry to try to link these, imaging mass spec discovery-based platform with specific cell types. What types of questions might we come up with that we think we can answer through a very directed and pointed collaboration?
I think one of the cool things we could address is how the expression profiles actually translate to produce metabolites, because there's not a good, really direct line between the metabolites that are produced and the expressed genome. That would be one of the most straightforward questions to ask, is how expression profiles correlate or don't correlate to what's actually produced in the cell or present in the cell. Yeah,
and that makes the most sense. I know we've talked about that a little bit. Now the link there has to be kind of spatial information, right? Unless we do some sort of cell sorting.
Yeah.
Mike, one question that popped in my head as you were talking a second ago, what's the limitation on the number of pixels? I think you said there's 5,000 pixels in a given experiment.
Yeah, we can probably increase 5,000 spots to maybe 20,000 spots. And that's in a 6.5 millimeter by 6.5 millimeter area.
But what is the actual limitation there? Is it throughput, or what stops you from going crazy?
Oh, it's basically the manufacturing technology, the spots, and how basically it's the machinery that, you know, how you can actually put the spots down onto a surface. Eventually, we can overcome that. We know a way of doing that where it'll look more like a continuous lawn of oligos.
Mm. Cool. Okay, so we have one kind of how we might question set up now for linking imaging mass spec to transcriptomics. But one thing I would like to circle back to is linking imaging mass spec, identifying cell types based on immunofluorescence, since that's a little bit more about what we're focusing on right now. Now, the way we're doing that, as I said previously, is, you know, we kind of connect the two through spatial registration. We tend to perform the immunofluorescence experiment on a serial tissue section. And as Elizabeth pointed out, we're working towards developing Codex as a platform for doing highly multiplexed immunofluorescence. And what we think the benefit there is that if we can effectively label, you know, a number of cell types in our case within kidney, we can then take our highly extremely multiplex imaging mass spec platform that again is purely discovery based, and connect, basically find the ions that we observe in imaging mass spec that are most correlated with the specific cell types that are labeled by immunofluorescence on the neighboring tissue section. So it's really a question of registration. And then once we do that, once we register the two, then we can effectively, through segmentation, we can effectively create masks and allow us to extract pixels on the imaging mass spec side that correlate with specific cell types. And from there, we can do some neighborhood analysis and things like that. So for our team, Raf and Elizabeth, you know, can we come up with some, you know, what do we still think are the major challenges that, you know, establishing collaborations might help us with in this process?
Well, I can only speak for myself because this is sort of ad hoc, but I think for me, it's the granularity, like how fine do we have to go? How fine is HCA? I can't really look inside HCA, and I don't know what the level of granularity is that they're striving for, approaching, having currently in the bag. Because of course you want to go as fine-grained as possible, but I don't know what is, there's a lower bound to that. And that's what I was interested in. Like I want to see where HCA is at and where they plan to go within a certain amount of time. And if we can do the same thing on our side, then we can have a super granular as we can be, link up what we see in one and what we see in the other. That's what I was thinking.
Mm-hmm. When you say granularity, you mean in terms of spatial resolution, or, you know,
yeah, spatial resolution.
Mapping all the cells in a given, you know, area of tissue, or.
Yeah, I don't know what HCA has as like the goal. What's the goal? Like how fine-grained is the description for the cell? I mean, it's obviously down to the cell level, but I'm like, okay, you could potentially look out organelles. You could potentially need it just up to the cell type level and not to the individual cell. There's a whole bunch of, like, it's not clear from, so maybe someone from HCA, if they can just educate me, then I can work from there.
Is anybody from HCA on the call right now? I know a couple of people have jumped on here recently. doesn't seem to be the case. But that's
Okay.
yeah. Well, I think that is one of the issues that we have seen, in HubMap is to, we tend to focus more on cell type and substructure, and that's just because of the spatial resolutions that we typically operate at. And now we've done a lot better. All of our HubMap data is collected at 10 micron spatial resolution. So we can separate one tubule from the next, and we can even subdivide something like a glomerulus within the kidney on a molecular level. But we're right at the cusp of single cell resolution, but we're not quite there. We're not generating multiple pixels across a single cell. So there is a bit of disconnect between the folks that are trying to do single cell spatial transcriptomics versus what we're doing on the imaging aspect, which tends to be a slightly more, slightly worse spatial resolution.
So what is we were talking about the 5,000 dots and the 20,000 dots. What is the size of one droplet or dot we can sample?
Oh, it's 50 microns currently.
50.
Yeah, 50.
That's not bad.
No.
And is that something you can play with, or is that like locked in?
Yeah, we can go a little smaller, but that's about where our current product is. We might be able to cut that in half.
Mike, I may have missed this, but is there like a orientation that these spots are in? Is it kind of a grid based, or what does that look like?
Yeah, they're in a grid. Their pitch is 100 microns.
Okay, so the spot is 50 microns, but the pitch is 100 microns.
That's right.
So your pixel size is effectively 100 microns.
Yeah, I guess so. I mean, it's more like you're sampling the tissue.
Yeah.
You only see what's over the spot.
Right. Interesting. Is there there must be then some physical limitation in terms of being able to, in a perfect world, right, your pitch would kind of equal your spot size roughly. That's probably not practical manufacturing.
Yeah, that's right, and that's where we're headed. But we don't really talk about the roadmap so much, or like when that could happen.
Sure. Interesting. Cool. Okay, so well, I think let's try to. We've got another 15 minutes here or so. So maybe let's pick another topic to talk a little bit about. Why don't we talk a bit about quality control? Because I think this is something that's really important. And actually, Mike, some of your input here on how you all address this might be helpful as well. One of the things that is challenging on the imaging aspect side is that, there are really good kind of standard ways of doing quality control for an LC proteomics experiment, for instance. That's pretty well established and pretty well controlled. There's really no method currently available to perform quality control, QA, QC validation on the imaging-based approaches. And so this is one of the things that we identified early on within HubMap, is that if we're going to play in as part of a large consortium, that we need to get this under control and get this developed. And I think it's going to be really powerful. So what we've done is we've developed a new method for doing this and software to do this. We're actually working on trying to get this published relatively soon. But it's pretty clear for us, the laser-based approaches, the method that we came up with and how we could validate and perform QC. But it's not very clear how other imaging-based technologies or ways of doing spatially targeted mass spectrometry would do the same. So effectively what we do on the imaging mass spec side is we're collecting kind of grids of images, of small images around the slide. And by varying the pitch between the pixels, we get a sense of how good our spatial resolution is, actually experimentally, what is our spatial resolution? What are the limitations? This approach gives us a sense of how our system is operating in general, how sensitive we are, how well the laser's focused, how consistent it is. And because we're using kind of internal standards over the whole slide to generate these grid patterns, we also then have built-in internal standards to compare intensity and allow us to normalize from one sample to another. So we think it's really powerful and should be a game changer in terms of being able to kind of deploy imaging mass spec to programs like HubMap. Now the challenge that comes in is, how do we do something similar for, let's say, a liquid-based approach? The questions are a little bit different there, but effectively what it comes down to is, how do we measure how well we're sampling the surface, in this case, using liquid droplet approaches? And so I guess, Mike, on your side, how do you all run QC on your platforms? And aside from kind of the analysis part, like how do you QC the sampling part of it?
So I hope I'm answering your question, but what we do is we ask our customers to use what we call an optimization chip, where they permeabilize a sample, and then the RNA is extracted onto the surface, and then we just produce a fluorescent image that tells you how much RNA is available from that particular sample. And so if you don't see anything, then you know the sample's been destroyed, and to take a different slice.
Now, is there any way, like in your setups, and I don't know your setup very well, but like on our case, if we have this liquid droplet and we don't make contact very well, we're going to get pretty poor extraction, and so that'll really affect our signal. Is there ways that in your setup that extraction process goes awry, depending on how the sample is, let's say, section thicker or whatever, it's at the stage is at a little angle, or what? I don't know how the setup is exactly.
Yeah, definitely, it has to be optimized. So we that's one of the things is we ask people to try different thicknesses, anywhere from 5 to 20 micron thickness, and then we try different permeabilization agents, and different amounts of time to actually permeabilize the cells.
Interesting. Yeah, so that's
I have a quick question.
Yeah.
How do you permeabilize the cells in the 10X system? Do you just use a detergent, or?
We use enzymes.
Like what enzymes?
We switch around, and I think right now we might be using proteinase K. I'm actually not in charge. That is not my product, but I know we at one point we were using proteinase K.
Thanks.
So I just got a message from Maggie, and I had the schedule a little bit wrong. It looks like we are tasked with taking a 15-minute break right now. So I want to make sure everybody gets their break, because I know this is a long day. And then instead of going to a plenary like we did last time and circling back to this, we're just going to take a 15-minute break and come right back to this subgroup and continue the discussion. So
If it feels like a good time to break now, if you're on a roll and you want to go 15 more minutes and then take a break. But what we wouldn't want you to do is go the whole afternoon without a break, because it's so important.
That's a good idea. Maybe let's finish this QC discussion, and then we'll just take a break since we're kind of on our own schedule.
But please make sure you do. Okay, I'm
We
going to
will.
check
We
on you.
promise.
If you're not gone in 15 minutes, I'm going to kick you out.
Absolutely. I like it. So I guess, for us, again, we think we've got it under control for the MALDI standpoint, but can we think of a collaboration that we could do to help us with QC for the more spatially targeted proteomics approaches, like these, like the liquid extraction surface analysis, or to speak for some of our colleagues in HubMap that are doing kind of liquid-based extraction? What kinds of collaborative projects can we think about, or questions that we can ask to help with QC more generally?
Well, Jeff, I have a couple things that I'm looking at, right? Well, that I'm interested in exploring a little bit. Like, what you have, QC is everything and nothing, of course. It's there's many different things you can control for. So my initial feeling would be to how can we sort of streamline the QC for different technologies across samples. So if you have multiple tissues, how do you capture and control for the variance between samples? And then within a sample, the two angles that spring to mind are, on the one hand, the content or the chemical angle or the spectral angle. Like how do you QC that the content is where it's supposed to be, or in the dynamic range where it's supposed to be, so we can have a signal that is comparable, or at least fixable if it is somewhere off to a side. Like if everything collapses to almost zero, there's no fixing it. So you have the content side, and then you have the spatial QC, because the more fine-grained our labeling becomes, and the more fine-grained our masks become, let's say, okay, cell type A is up there, the more fine-grained also our certainty needs to be, that when we say to the computer, in most of the cases, go to that location, that it ends up exactly sampling that location. So I look at it from a couple of, I don't know which one would be the more, my first instance would be, if we can't control a cross tissue, well, what's the point then? So that would be the most important one. And I think given that we are supposed to map, I would put spatial, well, maybe third, maybe second. And then, well, the chemistry, yeah, if you have no signal, well, there is no
yeah, well, that's
point, huh?
good.
So I don't know how others are doing this. Like spatial QC for us means the whole business of, we also use fiducials, but we sort of internally rectify with autofluorescence. QC, the spatial QC should also involve, and this is something we are working on for mass spec specifically, but I would be interested to see what others are doing. Hitting a target at a particular location. How much spread is there? If you say you're going to hit X and Y, how much plus or minus is that then that you physically get? Because it's never perfect. Right. And I have no idea how that is for example, something like 10X or any of the other technologies that have been looked at.
Well, as you know, on the imaging mass spec side, or just mass spec side in general, we approach, at least the comparison from one sample to another. You brought up a lot of points there. Well, and some of the spatial things too, but we kind of approach this from kind of classical analytical methods, if you will. So, if we don't have an internal standard, we use things like total ion currents. We look at kind of the total population of ions that we're detecting from one sample to another, or possibly one pixel to another, and then we can use that as a normalization factor. Or probably a better approach is to use an internal standard that is present at a kind of set concentration that is more or less at the same concentration at every pixel location, and then ideally same concentration from one sample to another. And we use kind of we do everything kind of relative to that internal standard to reduce any sort of variance from one sample to another, one pixel to another. I guess on the transcriptomics side, maybe Cody, you can speak to this too, or Mike, how is normalization done within the transcriptomics community from one sample to another? That's something I don't have a good handle on. Or, how do you deal with this kind of variance?
Yeah, as far as single cell data sets, there's a lot of different data integration or batch correction methods. And I imagine people are starting to apply these to spatial data sets as well, especially when integrating the single cell and the spatial transcriptomic data sets. But I think the references are kind of different than what you guys are dealing with for the most part.
What do you mean? Can you expand on that a little bit?
Yeah, I mean, just like the dimensionality and the information that you're working with from one data set to another might be different, and the way that you can control, for instance, capture efficiencies with mRNA are probably a lot different than the instrumentation that you guys are using with your mass spec.
Yeah,
and just the noise that you expect to see from one run to the next. But I'm not too familiar with.
Yeah, on our side, but I guess on the sequencing side, like how do you know, like right now on the sequencing side of things, if you run a sample, how do you make the call whether it's good or bad?
So I can speak to single cell because that's what I've mostly seen. For the most part, the biggest drawback to the single cell RNA sequencing is that you get a lot of ambient mRNA. So based on your dissociation, you have transcripts that are floating around that end up in every droplet. You encapsulate those, and you have this background. So basically being able to determine between the distribution of real reads that represent cell type that was encapsulated in that droplet versus reads that might be present across your entire data set. So for us, a bad data set would have a lot of ambient mRNA. And I imagine with spatial transcriptomics, you know, Mike mentioned like the different permeabilization times. If your mRNA is just diffusing all over your chip and you're just capturing it everywhere, then the signal to noise ratio drops dramatically. So like he said, with the optimization of that, permeabilization time is really important. And it's probably pretty variable from tissue to tissue, sample to sample, run to run, in other people's hands, things like that. So those are things to look out for.
So I know one of the things, at least what I've heard about from collaborators, is the use of like housekeeper genes to like normalize expression profiles. Can you speak to that at all?
Yeah, I mean, that's not something that we tend to do with our single cell data sets, and that's only because the ambient profile that we see from run to run is typically very different. So it's usually not the same exact set of genes that you can count on to normalize your data set or to use as a reference for what is noise. And it's really data set specific, which is why like some of the data integration techniques like Scanorama and Harmony and Combat, those are some single cell RNA sequencing batch correction methods. They kind of build models across data sets to correct that background and try to co-embed the data into a common space, if that makes sense.
So I have a limited transcriptomic experience, so this might be a naive question, but how do you then differentiate the ambient noise that's changing across samples from the biological, biological noise or biological significance?
Yeah, well, that's the trick. And so I think like in terms of quality control, if you have a data set that has an ambient profile that's cell type specific, then you're going to be confusing real cell identity with background noise. And in our lab, we've had kind of some data sets where we've had to go back and think about just the dissociation protocol and like kind of the tissue prep on the front end to mitigate cell lysis that would kind of convolve your distributions of like real cell identity with background noise. And, you know, as long as you can differentiate like a bimodal distribution of true expression and noise, then you're good to go. But it is data set specific and tissue specific.
Rolf, it seems to me that some of the challenges that the sequence, or some of the tools that the sequencing people are using to determine signal noise and differences in signal noise would be applicable to the mass spec side from a biocomputational standpoint and statistics standpoint. Is that right?
Yeah, I think so too. I think, so I'm not familiar with the types of signals that roll out of these technologies. Like, is it like a counter-based abundance, or is it something qualitative? What should I think of when you say, okay, we found this transcript?
Yeah, it's so it's usually based on unique molecular identifier counts. So for single cell RNA-seq, you'd have counts of each unique transcript that were detected in that droplet. For spatial, you would have it at each spatial transcriptomics locus or whatever coordinate dot.
So just to clarify, if you're going to do a batch of these things, you are you were saying that you take multiple samples and then you generalize models over that. But do you bring all the data in line? Do you pre-transform it and then you run a model, or you show it enough models? You show it enough unfixed examples until the model figures out, okay, this is the part that matters.
Yeah, I think typically like pre-processing is pretty standard in terms of like just normalization and transformation to go, I mean, for transcriptomics, you're dealing with like negative binomially distributed data, lots of zeros. So typically you would want to like normalize and then transform it with like a log or a hyperbolic arc sine or something to get it into a more amenable space, and then zero center that, but scale it, something like that. And that you would usually do beforehand, I think, and then kind of trying to resolve the batch effect noise. It builds the models on top of that normalized data to get them into a similar space.
Well, I think one area that we might want to take a look at is where these pre-processing methods can cross-inform on each other. Because if you have non-biological variation that you see in one imaging modality and non-biological variation that shows up separately in another imaging modality or another modality in general, that non-biological variation can have many sources, but if it's two different technologies, they might cross-inform on each other. Like you can have a part that is common, that is perturbing both the proteomics and the transcriptomics, and there might be a part that is transcriptomic specific, and there might be a part that is proteomic specific, and it might have a stabilizing effect to look at the I'm seeing lots of messages come by, to look at how these pre-processing methods can cross-inform each other. Because if you detect non-biological variation in one modality, you're sort of stuck with whatever it is you're running with. But if you can cross-cut it with the observations from another modality that are also trying to gauge what non-biological variation that is, that might be very informative. I think that will be a cool project.
So I see Maggie jump back on, which is why I kind of laughed there for a second, Ralph. So why don't we tie a bow on this? And I can think of two how might be statements that we can add to the end of this discussion based on what we've just talked about. And the first is, I think, and correct me if I'm wrong, is how might we find common biocomputational tools to help identify sources of variance between mass spectrometry and other technologies? So things like sequencing, and we didn't talk any about it, but potentially immunofluorescence and things like that. And then the second how-to statement, does everybody agree with that? Does that seem reasonable? Yeah. And then, so the second how might we statement is how might we design a collaborative experiment between, well, a lot of our group is on the call, but, HubMap and other consortia to identify like whether or not there are common sources of variance that are detectable between the various modalities. And one of the things that we can talk about in the next session is that comes to mind, though, is if it's a common signal, I would think that it's more likely that it's biologically relevant. If it's matching up across many modalities, then it's probably. I would be curious to get everyone's thoughts on whether or not we can distinguish a common signal that crosses modalities as a biological source of information, or if how we can.
Cutting artifact.
What's that?
A cutting artifact.
Yeah.
Or any other, like it can still be it can be non-biological and cross-cut different modalities if it's somewhere very much upstream in the sample prep.
Okay, so let's take a break there. Thank you for that discussion.
There was the question of how do we find out if there's correlation between metabolites and transcriptomics?
Yes. Elizabeth, did we add that above?
What do you mean above?
I think that question, Mike, that how-to question was added to the cell type discussion. Is whether
we added that above. I can highlight it.
Yeah.
I just was
you're doing a great job of note taking, Elizabeth. Thank you so much. So yeah, we can just highlight that. But Mike, that one's on there. That's a big one. That's one that we're very interested in as well. Okay. Well, why don't we just go ahead and get started? And
can you hear me now?
Oh, there we go. Now I hear you. Perfect.
Yes, hi. This is Pothur. I'm at NIH. I coordinate the transformative technology development initiatives. I'm at the NCI. I lead a group in the genetic epidemiology.
Great. Thank you very much. Sorry about that. It's always awkward on Zoom to know whether it's your technology that's failing or whatever. So okay, great. Well, let's circle back to our Google Doc, which is going to kind of help us navigate this. So, I think the idea here is that we spent the first half of this setting up what the problems might be that we want to address. And the second half is now to try to, as best we can, make these things actionable. So can we, put a point on this a little bit so that we can try to move forward with some of these things. And this is probably good because we've been talking about this, well, I know within our group and then within HubMap more generally for a bit. So I think if we can come up with some real strategies for accomplishing some of the goals we just talked about, it would be very helpful. So just generally, the things that we're looking to accomplish here is, give our collaboration a descriptive title. That we can do, that's no problem. We need to really identify the problem that we think we can solve as granular as we can, and then start to map out specifically what experiments can we do to solve the problem, to deal with the challenges that we set forth, and then importantly, make sure that we identify the people that we would want to be involved in, and what types of additional expertise that we need to kind of onboard to make this all possible. And then, hopefully be able to identify a meeting. Now with the group that's on the call, it's mostly our team and Cody. I know more or less where you live, so I can hunt you down. So we could find some people, but I think we also know at least on the HubMap side, the community of folks that would be interesting to pull into this, as well as some of the other people at Vanderbilt. So maybe it's a good idea to try to do some of those things. So why don't we start? The first problem that we can talk about is, just going in order, is how do we connect the kind of downstream metabolites and lipidomics to specific cell types. That was something that came up earlier. So that's the high level title of what we're trying to achieve is, how do we connect these downstream metabolites and lipids to specific cell types? What are let's first just list off what are some of the primary problems. Let's try to summarize that. Can we come up with 3 primary problems in making these types of connections?
So I'm guessing that you guys are primarily doing single cell segmentation and then quantification of MXIF to determine cell type. And then when you're mapping that to your IMS, you're kind of using those masks.
Kavi, you
serial
that a little bit?
Yeah, so yes, and a part of it, so we're not just going towards single cell, we're also doing single substructures. Just because within a lot of our work is within kidney right now. And so things like different proximal tubules, for instance, would cover more than one cell type or more than one cell. And so from that perspective, we're doing kind of larger scale. But yes, to your point that we're using, right now, we kind of have a single channel approach, so a single marker, just because that helps us reduce any false positives that we might have when we do the segmentation. So we have that kind of in place. So that's only using one marker, one antibody to delineate a cell substructure. And then from there, yeah, you're exactly right that we'll have the X and Y coordinates and we'll be able to go and do some downstream data mining on the IMS data. Does that kind of answer your question?
Mm-hmm.
Ruff, I saw you nod your head like you wanted to chime in. Did you have anything to add to that?
No, I mean, Kavya did a good job, so I'm totally on board.
Okay. Elizabeth, let's just kind of go around our group since we're here. What do we think are the primary challenges that we face in trying to make this happen?
In connecting downstream metabolites to cell type?
Yeah.
I think one of the major problems is a lot of metabolites are actually shared between many cell types, and the cell type defining markers are like what would be different between cell types is not as large as it is if you look at other types of molecules. So like proteins vary wildly, but most lipids are actually shared amongst different cell types. And so of the hundreds that are different, only like a couple are actually exclusive within one cell type versus another, and most is like relative abundance differences. So I think that's a challenge. It's not as defined as some other molecules.
Sure. Well, and you know, part of it though is I think if we're trying to make the connection, I guess one of the questions I'll throw back at you is, do you need, like, I can see that being a major problem if you're trying to use mass spec alone to define a cell type. You know what I mean? Like that's where that would be a real issue. But in this case, it's probably okay, right, to have a molecule span multiple cell types, and as long as you know that, right, and start to get a feel for you know, abundance differences in the different cell types is probably is what you're actually going for, not necessarily kind of on-off switches between cell types.
Yeah, it's mostly fine. I think it just depends on the goal. So like, if we have, so I've done, my backgrounds, for those who don't know, is in single cell mass spec. And so in some systems, on some instruments, if you don't have sufficient resolving power and sensitivity, all your cells actually look the same because so many of the large metabolites are actually shared. So like PC320, if you analyze, I think I looked at like 200 million cells, 99.9% of them had like PC320 in varying abundances. And so, and if you looked at it on a MALDI-TOF, they, like 90% of them had like basically the same profile. Now you move to an FTICR and they're like drastically different. So I think it's something where it doesn't intrinsically matter, but like it can get challenging if they all look the same and they're not supposed to. They're also the most abundant ones, are the ones that are shared, which just makes intrinsic sense.
Well, there's also the aspect, well, if you look at purely qualitative, then if it's just like, okay, is this molecule present or is this ion present or not present, then you can't tell. You could mathematically work it out backwards if your abundance was a good reporter. Like if you have, you can sort of use abundance to put some preconceptions on structure of which ion could be reporting which. But I'm thinking more of fragments already, so it's maybe a bit too far. I'm not sure, Jeff. What are we supposed to then come to a potential nucleus of a collaboration, or what is the?
Yeah, so I mean the idea is to yeah, exactly, form a nucleus of the collaboration. In this case, because of the folks that are on the call right now, it's probably easier for us to just establish what experiments we would want to do in an idealized case. And then we'll have to kind of hunt for our collaborations in post, since our group has shrunk dramatically after the break. Which is fine. I think it's good for us to brainstorm and identify what the idealized experiments would be. So I guess, we know, on our side, just within our group, we're using, immunofluorescence to make this connection. And so I think we have a good sense of that idealized experiment already. And that's basically, performing the experiment on serial sections right now, because we want to make sure that the imaging quality and the data quality is as high as possible. So we don't want to try to do it on one tissue section where both the imaging mass spec and the multiplex immunofluorescence might suffer. But so in our case, the idealized experiment is to collect a common modality between our individual sections so that the registration becomes as good as possible in connecting those two things. So we use autofluorescence to do that. And then we collect imaging mass spec on one and then multiplex immunofluorescence on the other, soon to be codex down the line. And use those immunofluorescence signals as effectively, a lighthouse for a specific cell type. And then that will allow us to then connect pixels in the imaging mass spec to specific cell types in the MXIF experiment. So I think we have a pretty good handle on that. I guess for our group, Kavya and Elizabeth specifically, in terms of that experiment, is there any shortcoming that you see or
Something to think about that hasn't come up a lot in our discussions, but came up a lot as I was trying to do this on the single cell level, because I used to make chemical profiles of like astrocytes and neurons and oligodendrocytes in the brain and different brain regions.
Mhm.
So like MXIF markers, say you have one that are supposed to stain astrocytes. On a good day, it doesn't stain close to 50% on astrocytes, and it's like the gold standard as an astrocyte marker. And so you have to end up using like overlapping markers to actually get a total number of the cell type. And then sometimes the markers won't work depending on like the state of the cell. So like if a cell's like about to die or something, some of the markers no longer work. And so that's, I don't want to say it could be a challenge, because you have to end up using like really multiplexing or really understanding the biology of the markers to get really good, confident profiles of your chemical profiles of your structures.
Mhm.
Just because the markers in and of themselves are chemicals that vary quite a bit. And usually the cool ones that like mark subclasses of cells or subcellular, or not subcellular components, subclasses of like major cell types like neurons, those antigens vary even more than like structural ones like GFAP, which still vary quite a bit.
Do we have a handle on the specificity of like?
Depends on the
Okay.
antibody.
Yeah.
Yeah.
Depends on how well validated it is.
So,
But I don't mean the specificity of the antibody. I mean the specificity as to saying that is cell type A.
It's very, very bad. So I was going through some of the markers we went through, and they stain lots of things, and like the lot to lot variability is really high for what markers. So I went back through and I'm looking at the ones we purchased again, and there seems to be a lot of agreement in the literature that publishes with them about what cell types they stain. So it'll be better, but they're still going to stain 3 or 4 populations, not just one.
But I think, you know, the approach of using multiple markers is ultimately where we want to go, and which is why, whether it's MXIF or Codex or any of these things, you know, having multiple modalities to get at the problem is probably for the win in this case.
But how do we encode for that? How would you say? So right now we have one immunolabel saying it's cell type A.
Yeah.
And that's and so we feel good about saying that's cell type A. Or do we feel good about saying that's immuno bloody blah and presumably tentatively assigned potentially as cell type A?
So when you go to like publish these results, I can actually share one of my publications. So when you go to like do this and you go to report these, you actually say, I'm defining this structures that are defined by being GFAP positive. In most cases, these are astrocytes, but you label it by the antigen you're testing for, not the cell type. And so you can have in the discussion like, oh yeah, this usually stains for this, but it also stains for X, Y, and Z. But the most appropriate way to say is these are this antigen positive and not actually declare it a cell type. And that's usually how it ends up being portrayed, unless you have like transcriptomics or something, or again, like 3 or 4 markers, just because you usually end up staining 3 or 4 different cell types with a single marker, just because they get shared a lot.
Well, and in a perfect world, you combine, I mean, it's, you know, again, it kind of circled, and it's why we're all kind of working on this is, you know, a multimodal approach will ultimately get you to where you need to go. And, you know, down the line, if we can start to, you know, coordinate imaging mass spec with some of these other technologies and figure out which molecular profiles, you know, correlate with a specific cell type based on a set of markers, then you can, you know, even start to use the lipid profiles or metabolomic profiles to help make that call on what the cell type is, and hopefully what the cell type is and subtypes and things like that. I think that's what we're all hoping for. That's all we're all working towards.
Yeah, and defining what cell types are is also an interesting problem.
Yeah, for sure, absolutely. So it sounds like, maybe one of the early things that we need to do is have good definitions of a test cell type. And, just thinking about, questions that we want to answer. So one of the things that I don't think necessarily we're the group to answer, this is probably, in our case, we need to, have a conversation with some of our kidney experts that are part of HubMap, but also with, some of the KPMP folks, is what is the best defined set of cells, in terms of immunofluorescence. Like if we had to choose 3 cell types to go after as an initial experiment, what is the best 3 to start with? And so KPMP is probably the place to start with that, just because it's kidney and they've spent a lot of time thinking about this.
And what do you mean by best defined? You mean best defined by people, or you mean most specifically labeled by?
Yeah, like what's most specific, what's been most validated, if we could pick a cell type to go after to demonstrate this, we'd want to pick cell types that were the best, options would be the most highly validated ones that are the most specific for a given antibody or antibody-based imaging, technology. Now, Cody, on the transcriptomic side, you don't really have this problem, right? The problem you have is you end up with all these transcripts and then you have to do whatever, TISNE or whatever, to figure out what cell types you have. If you don't have good separation there, then you don't define all your cell types. But
exactly,
it's a
yeah.
different problem for you, right?
Yeah, so from our perspective, with the single cell transcriptomics, you can kind of define cell types de novo with each data set, right? And that becomes your ground truth.
Yeah.
And so what I've been thinking a lot about in terms of integrating the spatial transcriptomics is using the single cell reference to deconvolve the admixtures of cells that you would expect to get at each of those spatial transcriptomics dots. Like Mike was saying earlier, you have somewhere between 5 and 20 cells at every location. So when you reconstruct your image, you don't have single cell resolution yet.
Mm-hmm.
And in that same way, I don't think that like image segmentation from the same slice, if you're doing MXIF on that slice before you do the spatial transcriptomics, that can't really define your single cells either, because you're going to have 5 or 10 cells within each spot, but their transcriptomes are just being captured and then registered to that area.
Wait, I may have misunderstood. Did you say you can't get single cell with MXIF?
No, with the spatial transcriptomics.
Oh, sorry, I misunderstood. I was like way confused. Okay, thank you.
Yeah, but ideally you could do MXIF on that tissue slice and then capture the mRNA in an untargeted fashion for spatial transcriptomics and then overlay those images, right? But your spatial transcriptomics regions are, 50 microns in diameter with 100 microns between the centers of each dot. So that's the spatial resolution you're working with. And then on top of that, your cellular resolution, so to speak, is a lot lower because you're going to have 10 to 20 cells at each of those, making up each of those transcriptome dots. But yeah, like Jeff said, like defining the cell types is easy to do with the single cell data. So if you have that matched, then that could be a reference there, which could get you into the spatial domain, which if you had registered proteomic information from MXIF or IMS, then all of a sudden you can link those directly.
Could you go back for a second? I was confused with the ground truth. You were saying that you define that de novo ground truth for every data set. Could you explain that a little bit more? Is it like you're specifying with a number of clusters, or how is that happening?
Yeah, so just through unsupervised clustering, you can annotate cell types based on their transcriptomes.
So you're just, would you separate like k-means clustering where you're saying that there's 10 cell types and separate the data, or?
That's one way to do it, yeah. More popular methods these days are like graph-based. So you basically build a nearest neighbor graph out of all your observations in your data set. So if you have a 10,000 cell data set, you create a graph based on the transcriptional profile of each of those observations. And then you can use like the Louvain or the lead-in clustering algorithms on those neighbor graphs to determine the optimal number of clusters, and then do differential expression analysis to determine, to make cell type IDs out of those.
Okay.
And so I think that that's, that's the most powerful part about the untargeted transcriptomics stuff, is that you can kind of build out your reference with varying levels of granularity, because you can make the clusters coarse, or you can make them really fine tuned, and then look at what transcripts are differentially expressed. And then based on those and what you expect your cell types to express, you can define what cell type they are, what state they might be in.
And you define the cell type. That's the part that I'm missing. So once you've done the cluster and you define the cell type, is that, do you go back in and match the transcript to the reference genome, or?
Yeah, I mean, you would just be looking for expected expression. So like in the colon, you have goblet cells that express mucin 2 and reg4 and CLCA1. So if you see those 3 transcripts as differentially expressed in one of your clusters, and that's probably your goblet cell cluster.
Sure.
And then like that, any cell that was assigned to that cluster based on your algorithm gets that cell type ID.
Okay.
One thing that I've always had a question about, Cody, so when you do this and you cluster the data in a way, are there standard ways of saying, like, I feel like it's got to be difficult if you have like a subtype or like a rare cell type or a new cell type, defining that within the data set. Like I always look at those plots from the sequencing data, the Tisney plot or whatever, where it's all clustered and they're all color coded in a very specific way. And I think to myself, like, how did you color code that? Like, that kind of looks like there's a little bit that might be breaking off from there, but you know what I mean? Like, how do you define how many clusters or how many cell types are in the data set without prior knowledge?
Yeah, and that's the other thing that that I've been thinking about a lot. And so recently people have been using more like matrix decomposition methods. So non-negative matrix factorization is a way to, instead of hard clustering your data using something like k-means or like the Louvain graph-based clustering algorithms, you can kind of assign continuous scores for different gene expression profiles that are defined by positively co-expressed modules of genes. So it's essentially like a principal component analysis. And you define your, what's the word? You're estimating your entire counts matrix as the product of 2 matrices that are basically loadings for different gene expression profiles, and then usage scores, or like the amount of each of those profiles that's expressed by every cell. So in that way, it's kind of a more continuous, you can have cells that have high scores in a couple different profiles, and these profiles, and this is completely untargeted and unsupervised, but they end up being pretty biologically interpretable. And usually you end up with cell identity gene expression profiles, and then cell state gene expression profiles that might be, different parts of the cell cycle or cell death programs, things like that. So if you have a goblet cell that's dying, then you would expect high usage scores for gene expression profiles that describe both the goblet cell lineage and cell death programs.
How reproducible is that? Like, if you do a triplicate analysis, do you get the same answer every time? And how do you how do you compare between?
Yeah, so methods like NMF are not deterministic, and they're stochastic. So there are a few different ways to overcome that by running a bunch of simulations and then building a consensus out. So there was a paper late last year in eLife called Consensus Non-Negative Matrix Factorization, and that's what I've been using. So basically, you do NMF a bunch of times and decompose your matrix, and then you kind of throw out the outliers and you come up with like the middle of your distribution, basically, that describes both your gene expression profiles and their scores and all of the cells in your data set. But I think one of the interesting applications for that would be doing it across data sets and then across modalities. So for instance, if you could use NMF to deconstruct a single cell RNA sequencing matrix into gene expression profiles, and then apply those profiles as ground truth to a spatial transcriptomics data set, then you could basically assign scores to each of the loci in your spatial transcriptomics slide that might tell you the makeup of different cell types or cell states at each of those regions, which again are made up of several cells. And ideally, any data set that's matrix based, you could apply this to. So you might even be able to do that across modalities. If you guys have, quantified MXIF data, where you have protein quantification from single cell segmentation, you could probably do some kind of decomposition of that as well. Those features.
So Cody, how do you do that across different tissues? Do you run NMF on every tissue individually, or do you run it on one big data set that you combine in some way?
So far, I've only done it on single tissue data sets, but it's been done on like whole embryo developmental data sets where you get a bunch of different tissues, and it should behave the same. I mean, it's essentially like a soft clustering. So instead of defining hard clusters and looking for a differential expression, which might bias those statistical tests, it gives you a more continuous score for different components of your data set, or meta features or meta genes. There's a lot of different ways to think about it.
Well, I was thinking more of it, if you have a certain cell state, and it's reported, if you do the NMF on one data set specifically, one tissue section specifically, and say, okay, this thing is state X. If you run it on a replicate, which is slightly perturbed, not exactly the same, how do I find that state again?
And that's a good point. I think that, I guess any gene expression profiles that you expect to see from one data set to another, they have to all be there or not, which is a challenge. So if all of a sudden you had a new cell type appear or a new cell state appear, and then you're trying to decompose that data set using a reference that didn't have that profile, then you'd end up with some noisy usage assignments, I guess, or cell type assignments, which would be tricky. So that's definitely something to overcome. And I think that, like, in terms of data integration approaches that people have built out for single cell RNA sequencing data, things like Scanorama, the idea is that if you have overlapping populations, then it will identify those and correct for technical variation. But then any unique populations would be conserved in the two data sets. And that's when you're just kind of like taking two data sets and trying to co-cluster them in a low dimensional space together. But I don't know how exactly you'd be able to do that with like the matrix decomposition approach.
Is this similar to the stuff that Elizabeth, you had done not that long ago with a lipid? Like, it was lipid, so I don't know how comparable that is in terms of transcriptomics, because I know those are different, but I remember talking about Louvain clustering with you at some point.
Yeah, so what we did is we took, I think we used like 10 rats or something, and then ran single cells from multiple rats over the course of like 5 months to like measure how the lipid profiles changed as a function of date and time, and if our clustering scheme was like reproducible. And it actually works out really well. And we were able to find rare cell types and then like bulk clusters. And so it was a very interesting challenge to take like 300,000 cells and then find what most cells look like and the different cell types, and then some rare cells and then like partial cells. We then what we also did is we correlated our t-SNE and recolored based on ICC profiles. And so that's actually how we define cell type. And then found like cellular populations is with the immunocytochemical profiles, and then some PCA and Louvain-Jacquard clustering analysis.
And so Cody, is that pretty similar to the work that you were kind of proposing, in the sense that you could apply these same techniques most likely and then identify or be able to separate those, and then getting back to Jeff's point about making sure that they are more reproducible across samples?
Yeah, I mean, that sounds pretty similar. So Elizabeth, you were identifying cells based on their lipid content.
Yeah.
Like, what were the features of that data set? It was just lipid IDs?
Lipid
and
IDs.
Quantities?
We used quantities, but it's not like transcriptomics. It's not as quantitative as transcriptomics is. And so we normalized and used relative abundance to not, so I kind of looked at it like there's a couple lipids that are similar to housekeeper genes. And they're at similar abundances within all cells. And so we normalized to those.
Okay. And then presumably those features would be available in the spatial IMS data, so that you could kind of link those two data sets?
Yeah, we started.
If we're talking about creating a cell type reference and then trying to map that.
Yeah,
spatially.
so we started doing that. So what we did a long time ago, this was actually towards the middle of my dissertation. We did single cell analysis, and then did t-SNE and clustering, and then did IMS on a tissue that was actually close to serial to where we got our dissociated cells. So we took like a thicker section for the dissociate, so it wasn't like serial. And then we could actually match different regions of the hippocampus to the dissociated cells. And so the profiles actually were remarkably similar from clustering on the tissue to the single cell dissociates. But that project kind of died in the middle, which was sad. So yeah, you can. And so it looks like single cell data actually corresponds fairly well to the tissue data.
Okay, why don't we, so just in the sake of time, because this has been a really great discussion, but we've got 14 minutes here to kind of define this experiment. So can we circle back real quick and try to put a point on at least this one experiment in terms of figuring out, what we would want to do in the long run? And specifically, like I said, I think we've got a pretty good handle on the microscopy side of things, other than, figuring out the right combination. Well, I think one of the questions is on the microscopy side of things in terms of connecting imaging mass spec to cell types is, what are the best cell types / antibodies to look at that have been well defined by KPMP?
Would it be valid? So when you say the best, then is the best the ones that you have a unique label for with no other competing label? Well, no. Ideally, you would want, I'm trying to get to a definition of the best, because I can imagine that you, from a certain perspective, the best is the one that is covered by multiple stains, and you get confirmation across the stains, and they're all pointing to the same guy. But you could also say best in terms of not having that and taking up as little in the, if you only have 24 labels to put down, you don't want a cell type that is double dipping,
right?
You see, so
well, I think the best case scenario is, well, I think for an initial experiment we don't need every cell type, so we're just looking for 2 to 3 cell types that we can well define with 2 to 3 antibodies, essentially. Elizabeth, would you agree with that?
So I was actually going to jump in. when I think of like the best antibodies or like the best markers, you can actually look and say, how reproducibly does this stain X proportion of a cell population? And some vary quite wildly and some don't vary as much. So the reason GFAP is like the premier astrocyte antibody is they know exactly how to make a cell produce GFAP. So like if you have a cell line of astrocytes, they actually aren't GFAP positive despite being astrocytes until you feed them a specific concoction, and then they become GFAP positive. And so the biology or the pathway analysis of GFAP in astrocytes is super well understood. So you know when it's positive and when it will not be positive. And so I think that defines the best antigens to look for, things that are like really well understood, when you know it's going to be positive and why it's positive, and when you know it's going to be false negative and why it's falsely negative.
So, yeah, so where are you getting the measures for that? Like I understand GFAP has a high specificity, therefore a low spread on it getting it labeling a particular cell type. But where are you getting this data?
Usually, so the nice part about GFAPs, it's been published like a bajillion times. So usually you just look at papers and then like reviews do an analysis of like for this lot there have, and even vendors will provide this information. There's 200 papers that say that this antibody and this clonality and this lot stain X marker. And then other people have said, okay, this antigen is involved in this pathway. This is what goes into that pathway. So there's like, it takes a while to actually break down that literature. If you're lucky, there's reviews that do it for you. So GFAP is really common because it's studied a lot. There's some for neurons. So we just have to find those in the kidney, and I'm sure they exist. It takes an amount of work up front to find them.
You get to hear really highly specific labels.
Yeah.
The ones that have highly specific labels.
Yeah. And so the nice part is the vendors often will do that for you because they want to sell antibodies. And if something's been published in 400,000 publications, it helps them sell more antibodies. So it's a combination of knowing initially where to look and what you're interested in, and then knowing where to look after that.
One question that I have on that though is that, you know, that kind of validation that you're talking about works on cell lines, but it doesn't work in tissue.
Yeah. So you do the best you can. So it's one of those things where the problem is I know GFAP really well because that's what I did. But like there's a specific shape. So you have someone who really like knows the biology well that can say, yeah, this is staining the right thing. The shape is correct. It's in the correct region. So it's like an iterative process of you start off as strong as you can, and then you get someone to validate what you're doing. And you validate it as best as you can. So the nice part is some antibodies like just work remarkably well. And if you're not doing something crazy, they'll continually work. And then you just have someone check, yeah, this makes sense. This aligns with what we've gotten previously. This aligns with all the previous literature. The problem is where you get a finding that doesn't align well with the previous literature, and that happens more than you would want. Just because antibodies aren't very specific.
Right.
So Jeff, just up front, it might be good to tell the biologists, give us the most clinically relevant cell types, like make a top 5 or top 6, and then we can dial those down to the ones that we can find highly specific. So that way we can ensure that whatever path we go down, we can always tie it back to the clinical story. Does that make sense?
That's a good suggestion, to have someone say, these are the most important structures in the kidney. Not saying anything about like what's in those, but like, what are they? And then go back and find antibodies, because that'd actually be more straightforward than the.
Yeah, we can let the kidney people just say, what are your favorite cell types? I mean, it must tie to some particular disease having some onset or something like that, probably why it's their favorite, but and then we can dig down.
Okay. So, okay, let's just walk through. We need to fill out this final template here in the last 6 to 7 minutes. The title of this project is something like linking spatially targeted mass spec to specific cell types through IF and spatial transcriptomics.
Is this to serve as a new yeah, collaboration?
Well, it's kind of building off of what we're doing right now, I would say, just because we're all here. And, you know, ideally what we'll do now is, you know, like I said, everybody here, we all know each other already, but, you know, ideally we'll reach out to the broader NIH-HCA community to see who else might have samples that make sense.
Right. Okay. Okay. Yeah.
So problems that we think we can solve through this collaboration, I think, well, along the lines of what we were just talking about, identifying the proper test system.
What do you mean by that?
Well, just the combination of tissue, antibodies, like what would be the ideal experimental system that we would start off with, right? Thank you, Anonymous Cheetah, for fixing my typo. So what's another problem that we think we can solve? So with this on the transcriptomic side, Cody, what's one thing that we think we can solve by trying to identifying a common space to work on?
Yeah, I mean, for me, I think it goes back to correlating transcript abundance with protein abundance. We can do protein IDs in MXIF, but that doesn't necessarily correlate directly for that exact cell to the amount of transcript for the given protein. But if we can do spatially untargeted mass spec and transcriptomics on the same tissue, or at least the same sample with serial sections, you would think that you could connect those to identify cell types. And then I guess at that point, you're looking at like temporal differences between protein expression and transcript expression, right? Something along those lines.
Well, I think you can also look at it from the perspective of using both technologies to really, well, you could do cell type definition in one, cell type definition in whatever information you have in the other. Does it differ much? Does it cross-inform on each other? Mm-hmm. So even the, I mean, a very cold statement would be, oh, does transcriptomic only based cell type specification hold up in the proteomics realm? Like that would be a sort of an adversarial way to put it, but that sort of spatial specificity thing, yeah, that might work.
Yeah,
I guess which modality? Go ahead, Kavi.
Kavi, go ahead.
I was just going to say that kind of using one to reinforce the other and refine, and so really taking that multimodal approach to the extreme by saying, like, we found it by transcriptomics, but now then we used the lipidomic profiles and we kind of culled the data set or pruned it in such a way, or, you know, identified multiple within that one cell type. So kind of refining it from there.
Yeah, we're thinking about like, you know, which modality gives you more specificity or granularity in like your cell type clusters. Like you might be able to define broad cell types with transcriptomics, but if you look at those same cells based on their lipidomic profile, you might see some heterogeneity that could tell you a little bit more about that population. So yeah, like you said, Kavya, like kind of refining that information across.
Yeah, that's good. I think that would be a really nice outcome of this, is if we can compare effectively what you're talking about, Kavya and Cody, are comparing different modalities and molecular classes in terms of their ability to be specific and granularize the various cell type definitions that roll out of the platform. Yeah, I think that'd be really powerful. Now, in terms of expertise, so what I have is in terms of what we want to do is determine the tissue and cell types to start with, just a very practical thing. We want to collect the multimodal data on as close to the same tissue section as possible, but more likely are serial sections, and then compare and see, once we link the data sets together, how well can we define a cell type, and even better is like we just talked about, can we start to compare different molecular classes for their capability to differentiate these things. The last thing that we want to talk about, or that we should just circle back to, is what other expertise do we need? So we have the biologists that we need to get involved to help us identify the proper cell types to work with. Do we see any other expertise that we're missing?
Jeff, I don't know if you're including pathologists within the biologists. I'm assuming.
Yeah, sorry. I'm being very general and colloquial with my language. So yeah. So on our side, Agnes Fogo and Mark DeCaster and Ray Harris, and then obviously Sanjay Jain and some of the folks at KPMP, if we're going to focus on kidney, which I think we probably would. Cody, sorry, you're outvoted here, but so we'll probably start on the kidney side. But is there anybody else that we think we're missing in this little subgroup? Oh, sorry, I'm not seeing your warning. Perfect. Yeah, no problem. Thanks, Maggie. So we have only a few minutes here, so we're gonna tie a bow on this. Anybody else that you think we're missing?
No, I think, do you need to open it up to the HCA or to like, does this need to take the form of an open invitation for people to go like, oh, I like this, I want to jump in?
Yeah, that's something that I'll look into on how we promote this, or kind of throw it out there. I know how to do that within HubMap. I don't necessarily know how to do that kind of cross consortia. But I think at the very least, this might be a good jump off point for like our little group, kind of
definitely.
Vanderbilt to U Delft kind of subgroup to try to get the ball rolling on these things we've been talking about for a long time. Kavya's made a lot of progress on the segmentation work, and Heath and Kavya and Raf are working really hard on the integration of the imaging mass spec with immunofluorescence. So it might be a good opportunity, Cody, to kind of now get everybody involved and see if we can't start to really put a point on some experiments that we can try.
Sounds good.
Okay, the last thing that we need to throw on is just our next meeting date. Why don't we just shoot to have a call, at least with you, Cody, if not you and Ken, for maybe in a couple weeks. We're all sitting at home, so we can try to set something up. So why don't we just roughly just do 2 weeks from today, and we can adjust it from there. Sound
Perfect.
good?
Yeah.
Cool. Okay, everybody. Well, thank you. I know the group got really small at the end here, but it didn't seem too painful. So that's good. That I think that bodes well for potential collaborations down the line. That we're able just to kind of talk through some of these problems and make some progress. And yeah, thanks a lot. And I guess we're going to jump back to the plenary now. All right. Thanks, everybody.